Well, welcome everyone to a brief overview of the cells, synapses, receptive fields and pathologies associated with the outer plexiform layer. The outer plexiform layer of the retina allows transmission of visual information vertically towards the retinal output cells as well as laterally to other parts of the retina. It lies between the outer and inner nuclear layers and consists of synapses between photoreceptor, bipolar and horizontal cells. Photoreceptors respond to light and convert this to electrical energy through phototransduction. Rod photoreceptors operate in dim light conditions allowing for night vision, while cones mediate colour vision and visual acuity by rapid responses to varied light intensity. Adjacent photoreceptors are coupled via gap junctions, allowing both cone-cone and cone-rod interaction. While this coupling reduces visual acuity, it improves signal-to-noise ratio, which is important for rods in dim light, and it minimises redundancy, as photoreceptors that would otherwise be suppressed are used for other pathways. Bipolar cells are intermediary neurons linking photoreceptors to the output retinal ganglion cells. On cells are depolarized in light, and off cells are depolarized in darkness and hyperpolarized in light conditions. Morphologically, midget bipolars have small dendritic trees and receptive fields, while diffuse bipolars have extensive dendritic trees and receptive fields. Input can be from rods, cones, or rarely both. They project to different parts of the inner plexiform layer depending on their response type and input. Adjacent bipolar cells of the same type are electrically coupled, similar to photoreceptor coupling. Horizontal cells allow communication between adjacent parts of the retina and mediate centre-surround receptive fields. The majority contact cones and are tuned for either light intensity or colour, while those with rod connections are more sensitive to light. Like photoreceptors, horizontal cells are electrically coupled via gap junctions, but only between the same horizontal cell types. Photoreceptors synapse with bipolar cells and horizontal cells in the outer plexiform layer at ribbon synapses. Large numbers of glutamate vesicles are adhered to this ribbon, which releases glutamate via exocytosis and allows for sustained visual responses. On-bipolar processes extend into synaptic invaginations, while off-bipolars make flat contact externally. The synapse is glutamatergic. Off-bipolars and horizontal cells share ionotropic glutamate receptors, such as AMPA or kainate receptors, which open postsynaptic cation channels and are sign-preserving. In contrast, on-bipolars have metabotropic mGluR6 and LAP4 receptors, which are coupled by G proteins to the TRPM1 channel and supported by nictalopin. Glutamate closes cation channels and causes sign inversion. Some on-bipolars also possess ionotropic glutamate receptors which permit hyperpolarizing inward chloride currents. At rest, rods and cones are partially depolarized by inward cation currents, causing calcium influx and exocytosis of glutamate vesicles at their synaptic clefts. This depolarizes off-bipolars and horizontals and hyperpolarizes on-bipolar cells. With light, photoreceptors hyperpolarize and glutamate is reduced. This closes AMPA and kainate channels and hyperpolarizes off-bipolars and horizontal cells. However, the reduced mGluR and LAP4 signaling disinhibits on-bipolar cation channels and depolarizes them. In addition, inward chloride currents are stopped, aiding depolarization. Concurrently, as horizontal cells are hyperpolarized, they extend to surrounding regions to mediate surround inhibition. Feedback onto surrounding cones results in cone depolarization and increased glutamate release, essentially an off response. This is achieved either through increasing synaptic pH, creating membrane charge through dendritic microchannel currents, or by the removal of tonic GABA inhibition. Alternatively, horizontal cells may feed forward onto bipolars directly, however the mechanism behind this is less clear. This underpins the centre-surround receptive field of bipolar cells, where light falling on the centre of the receptive field will elicit a response that is opposite to when light falls on the surrounding region. This may also extend to colour processing, and the net result is a visual system which is designed to detect changes in light and colour by augmenting any differences between neighbouring retinal areas. When processes of the outer plexiform layer are disrupted, vision is affected. In melanoma, autoantibodies against the TRPM1 channel results in dysfunction of on-bipolar responses. This manifests in the absent B wave on ERG and symptoms of reduced night vision and hallucinations of shimmering white lights. Similarly, the majority of cases of congenital stationary night blindness are associated with defects in the on-bipolar pathway, either through mGluR6, TRPM1, or in the complete form, nictalopin. On-bipolar signals are thus suppressed, while off signals are spared, suggested by absent B wave but normal D wave on ERG. The predominant deficit is night blindness, likely due to the single on-bipolar rod pathway, while collateral signals and off-bipolar pathways appear sufficient for vision under normal conditions. The variety of bipolar and horizontal cells and their multiple interactions at the outer plexiform layer greatly expands retinal specialization and the parallel processing of multiple visual components. We have seen a brief overview of their interaction and associated pathologies. Thank you for listening.